Pablo, it's a great pleasure to be here. The last time I spoke in Chile was 20 years ago, I think, and it's been too long. It's very nice to be here. And also, thank you for doing this in English. My Spanish is not too good. I would have a hard time giving a scientific seminar in Spanish. It would be like kindergarten. I want to tell you about work I've been doing in my laboratory with a group that's been with me for a long time, and it's a body of work that's taken about 15 years to develop. And I'll do my best to show you the state of the art of where we're at with this. And I want to make sure that everyone can please have an opportunity to ask questions, because I'm going to be presenting a lot of different topics with different aspects of neuroscience, different types of analysis. And if there are any questions, it's much more interesting to ask the questions in the middle of the seminar than waiting until the end. So please interrupt if there's things that you don't understand. So this is a nice picture of Montana. It's up in the national park, Glacier National Park, near the Canadian border. And I hope to see some similar country in Patagonia. Anyway, I've been studying visual short-term memory or visual working memory for about 20 years or so. And I want to tell you about a body of work that's been ongoing for about 15 years. And the people that were working with me on this are Stephen Hoffman, Nick Dotson, and Baldwin Goodell. And Nick did his graduate work with me as well as a postdoc. He's now he went to UC Berkeley, and he's now at the Salk Institute. Stephen Hoffman, he finished his PhD recently, and the work I'll tell you about today was mainly his PhD thesis. And Baldwin Goodell, I've been working with him. He's an engineer, and he made a lot of this work possible because he's a very talented mechanical engineer that helped us implement modern engineering methods into the design of the hardware to run these experiments. And he and I are currently running a business as well that acts as a consulting company for other researchers. But yeah, I'll be talking mainly about the work that Stephen and Nick did. So I want to begin with a little bit of background about the organization of the cerebral cortex in the macaque monkey. And this is a picture taken from the work of Nicola Markov from a study in 2014. And I put this up here just to remind you that that the cerebral cortex in primates has been, over the course of 30 or 40 years, has been parcellated into a large number of specialized areas on the basis of histological differences, connectional differences, and physiological differences. And as of 2014, the state of the art in this parcellation was about approximately 91 cortical defined cortical areas, and they're color coded here on the right hemisphere of a macaque monkey. This is a picture of the right hemisphere. And if you unfold the cortex into what's called a flat map, you can label each of the cortical areas and see what their spatial relationships are here. So this would be, up here would be prefrontal cortex, orbital frontal cortex, the temporal lobe in here, the ventral temporal lobe down here, somatomotor areas in here, parietal cortex, cingulate gyrus in here, and then back here, the occipital lobe. So each of these areas have their own distinct functions and overlapping functions with adjacent areas. But one can see that function is widely distributed over the cerebral cortex, but it's also specialized, which is kind of an interesting problem to try to fit models or theories of cognitive function into this complex architecture of the cerebral cortex. Because doing something simple like looking at an image and making sense out of it, paying attention and moving your eyes, and then perhaps reaching with your hands to grasp an object, involves the coordination of many of these areas working together, and how this process works at a global level still remains one of the biggest mysteries in neuroscience, I would argue. Another aspect of this, and this problem certainly applies to something like visual working memory. Another aspect of this that also comes from this anatomical work, from the work of Nicola Markov, who did his thesis work in Henry Kennedy's lab with a large group of collaborators. They did some spectacular studies that are continuing to publish new results to this day, where they put what are called retrograde tracers into the cortex, such that the cells that project to the location where the tracer is injected would transport the dye back to the cell body of origin, and then they could histologically analyze the brain of each monkey and look at the origin of the cells that project to each location in the brain that was injected. And I'll just sum over a lot of work here and give you the punchline of this work. If you look at and this is talking about connections between neurons that are within the cortex, largely. So if you inject a point in cortex, for example, in area V4 or V2 or V1, which are the early areas of the visual cortex, what you find is that the cells that project to that location within the same area, they refer to those as intrinsic connections. All of the cells in the brain that project to the injected location lie within the same area. Something like 75%, 75 to 80% of the cells that project to a point in cortex lie within the same area. So they call those intrinsic connections. And then cells that arise from other areas outside the injected area, but from nearby areas, so what they call short extrinsic connections. Approximately 15% of the connections arise in adjacent cortical areas, and only about 3% of the connections that come into any point of cortex arise from long distance connections in the brain. So for example, from parietal cortex to visual cortex, or from prefrontal cortex to parietal cortex, or to temporal cortex. And interestingly, really very interestingly, approximately 1.5% of the connections to any cortical region arise in subcortical structures. So most of the connectivity is local, but there is a lot of long-range connectivity, but it's very sparse and it's very weak. And after a large body of work, they were able to count the number of areas that project to each of the areas that were injected with the dye. And this is a plot here, this is a plot here that shows the number of areas, cortical areas, that project to each of these individual areas. So for example, area F1, which is the primary motor cortex, it receives extrinsic connections from approximately 30 other cortical areas. But the frontal eye fields up here in the prefrontal cortex receive connections from a large part of the cerebral cortex, like approximately 80 other cortical areas will project to the frontal eye field areas. And on average, cortical areas that were injected, they receive input from something like 55 other cortical areas. But these connections are very sparse and they're very weak, and they fall off with a logarithmic function as a function of distance. So there's a high degree of interconnectivity across the cerebral cortex among all of these cortical areas, but that connectivity is weak and it falls off dramatically with distance. So the point of showing you all this is that there are all these specialized areas. We know that many of them have to be active at the same time in some coordinated pattern when you do a simple cognitive task.
Charlie?
Yeah.
Can I interrupt you?
Yeah.
This is very impressive in the way that it seems that most areas are connected to a lot of other, at least half the brain, right?
Exactly.
So what about the idea of hierarchical connections? I mean, and top down and bottom up. This seems to be not consistent with this.
Well, that's a very good question, because they have gone on to do a very careful analysis where they look at the cells of origin that project to each location, and they look to see what layer the cells are sitting in. And if the cells of origin that project to an area are up in the superficial layers, then there's a good argument to think that those areas are what referred to as feed-forward projections. So going from, for example, from V1 to V2, or F1 to F2, or area V4 to inferior temporal areas, for example. And then the cells of origin that lie in the deep layers are typically associated with what's called feedback connections. And if you look at the ratio of the cells that are in the superficial layers to the deep layers, this is a metric that they developed, you can develop a very nice hierarchical organization of cortical areas based on the ratio of superficial to deep cortical projections that come from a particular area. And that's a very sophisticated analysis that's been published and published and republished, but they have a very nice map now that ultimately relates back to the early studies of Velleman and Van Essen, where they did a similar sort of thing. So they do have this very nice hierarchical organization, and I'll tell you a little bit more about that later. So what this poses is a problem. Is like, how do all of these cortical areas communicate together to carry out some even very simple cognitive tasks, not to mention complex tasks? And I would argue that that's one of the big mysteries of brain function. But it leads to a question. So for example, if you're talking about something like a visual memory, a short-term memory, it leads to some very basic empirical questions, such as, what is the spatial, temporal, and spectral organization of cortical and subcortical neural activity states when you perform a short-term memory task? That same question would apply to any type of sensory motor or cognitive task. What is the whole neural circuit that's involved? And what is the spatial distribution of that circuit? How does it evolve in time? And what are its spectral properties? As measured perhaps in the local field potential. So very empirical questions that I think we still don't have an answer to. And then what are the patterns of functional interactions that underlie these states? It's also a wide open question, and partly because the neuroscience has been largely dominated by making measurements from one or perhaps a couple of areas or a small number of areas at the same time. So we have a very limited view of how all these interactions might take place at the global level. So big unanswered questions. In the case of visual memory, short-term memory, which aspects of the activity patterns relate to the storage or representation of memory, how you maintain that in the presence of distraction, and how you implement attention to maintain these memories, how you might retrieve memories when certain cues happen, and how these patterns relate to performance. For example, when you make mistakes, what do the activity patterns look like? And these are all very largely unanswered questions that we wanted to look at. So most areas have focused in terms of short-term memory in prefrontal and parietal areas, and usually, classically going back 30, 40, or even 50 years, all of these measurements have been measured in one area at a time, often one cell at a time. And it's only been in the last 5 to 10 years that people have started to look at larger numbers of simultaneous measurements to see how areas are communicating together. But to get a global picture, we still have relatively little insight. So I wanted to give you a little bit more historical perspective, because there's a long history of measuring global activity states particularly in humans, interestingly enough, that goes back to the 1930s and 40s. And then there's a classic paper published by Jasper and Penfield in 1949, where they laid out their methods for making multiple measurements of the surface cortical EEG in patients that were undergoing neurosurgery for epilepsy. And they developed this bizarre method to mount these, essentially these manipulators where they could place a contact onto the surface of the brain, and then they could, of course, measure epileptic activity, but at the same time, they could record the ongoing EEG and look to see if the EEG varied from one cortical region to the other. And there's a whole series of studies in the 30s and 40s and into the 50s where a lot of work had been done on this, but it had kind of been, fallen off or replaced by the single unit recording method in experimental neuroscience. And this work was interesting because they had, at the time they had no computers to analyze this, and they just would do hand drawings. They have polygraph traces of the voltage, and they would do hand drawings of the brain and basically say that, around the central sulcus, there was a lot of what they referred to as beta activity. So in the frequency range of 20 to 30 cycles per second. And then outside the central sulcus, they made these drawings of what they referred to as alpha activity, which was the first brain rhythm to be discovered back in the 1920s. And there's some interesting quotes. These are very interesting papers to look at. I recommend to take a look at these. So there's one quote here that practically the pure beta rhythm made it possible to identify the precentral gyrus even before its exact location had been determined by electrical stimulation. So they saw different types of patterns in the frontal cortex, in the sensory motor cortex, and then back in the parietal and visual cortex. And much of that work had been largely dropped. Was another group, Garvin and Amador, published a paper in 49 also that showed similar effects in the macaque monkey, in this case, monkeys under anesthesia. They made simultaneous direct recordings of electrical activity from cytoarchitectonic areas that showed a marked difference. So these were around the central sulcus. This is in inferior parietal areas. This is occipital. These are prefrontal areas. So some really good reasons to believe that the electrocorticogram varied from one area to the next. Well, in a very famous paper in 1978, Vernon Mountcastle published this paper called An Organizing Principle for Cerebral Cortical Function, the Unit Module and the Distributed System, where he basically proposed that cortex is essentially the same everywhere. And the way that cortex has evolved is by repeating or increasing the number of cortical columns that exist in the cortex. And that was responsible for the expansion of the neocortex in mammals, and particularly in primates, and even particularly in humans. And so there's a couple of quotes here that are worth looking at. He says, "I wish to explore the general proposition that the processing function of the neocortical modules is qualitatively similar in all neocortical regions. Put shortly, there is nothing intrinsically motor about the motor cortex, nor sensory about the sensory cortex." So basically he's saying that a column is a column, no matter which part of the cortex that you work with or you're looking at. And they're all essentially anatomically designed the same way. What makes them specialized is the type of input that they get. It's a bold claim, but very good reasons to make that claim. And then he reviews the earlier literature of what I just showed you and said that it was discovered that the EEG of very large regions, such as the frontal, parietal, and occipital lobes, are indeed quite different, but no characteristic differences have been found between records from areas within those larger regions. Until now, at least, study of the spontaneous slow wave activity of the neocortex has revealed little of its intrinsic functional organization. And then in conclusion, he makes a rather bold statement. He says, "In summary, I conclude that the cytoarchitectural differences between areas of the neocortex, which is of course responsible for this parcellation of these 91 cortical areas, mainly reflects differences in their patterns of extrinsic connections," which is what the Markov studies have so nicely and elegantly demonstrated, And he suggests that the neocortex is everywhere, functionally much more uniform than hitherto proposed, and that its avalanching enlargement in mammals and particularly primates has been accomplished by replication of a basic neural module without the appearance of wholly new neuron types or of qualitatively different modes of intrinsic operation. And essentially the short punchline of what I want to tell you is that we believe now that this last statement is incorrect. But there are many models that make use of this central postulate of Mountcastle. In particular, there's a nice book that you might want to take a look at called The Thousand Brains Theory by Jeff Hawkins that was published this year that heavily relies on this basic final conclusion here. But what I'm going to argue is that that's actually incorrect. So in order to get to that point, we want to know what does the spectral and temporal properties of the cortical local field potential, how do they vary across the cortical map? And how do they change during visual short-term memory? So very basic type of empirical question, but a hard one, a very difficult one to answer. And that's because the current methods that we have available to us that use the EEG or perhaps the ECOG methods in neurosurgical patients or the magnetoencephalogram or functional fMRI methods, they simply don't have the spatial or the temporal resolution to answer the questions adequately. So you need some new methods. So I would say about 15 years ago, we came up with the idea that we really need to develop a new method to sample activity over as much of the cortex as possible in macaque monkeys, and to do that simultaneously while the animal is performing a simple cognitive task. And we chose a paradigm of visual short-term memory, in large part because my work has always been in the visual domain. And we published the method study for this, and a follow-on study of the unit activity. And what I'm going to tell you about today is the patterns of the local field potential that we analyze from that study. So we use spectral power and coherence analysis, and I'm going to focus today on the spectral power aspect of things to look at the LFP, the local field potential, from each electrode and its pattern, its spatiotemporal organization. And ultimately, we want to look at the pattern of functional interactions in the form of coherence or phase coupling. But I'm focused mainly today on power because the coherence analysis is a work in progress, and it actually ends up being a much more complicated set of questions to look at. So again, please interrupt me if there's any questions that come up about this. The method was pretty radical. It was a pretty radical departure of the traditional method for doing electrophysiology in behaving animals. And essentially the idea was to put a large chamber that would span an entire hemisphere of the brain, and then to put into that chamber a custom designed microdrive that contained 256 independently movable electrodes. And so we implemented that method in two animals with some slight variation between the animals. And the second method, we got better data than the first. But the basic idea is that each electrode is attached to an actuator, which is a little screw-driven mechanism. So you can put a screwdriver in here, you can rotate the screw, and when you rotate the screw, that will cause the electrode to advance into the brain. And so over the course of weeks and months, you can insert all of these electrodes into different parts of the cortex and make repeated measurements on a daily basis that span months or nowadays even years. And the old method that we used was to machine these parts and then to assemble them together sterilely and then implant these into the animals. Nowadays, we're using 3D printed technology to build this as a single part, and it's making the devices much more reliable and higher capacity and so on. But I'll tell you about the results that we got from these experiments. So after these complicated surgeries and doing our best to make sure the animals are healthy and everything's working okay, we had trained them to perform a short-term visual memory task, what we call an object-based oculomotor delayed match to sample task. And it's very simple. So the animal has a computer screen in front of him. There's a fixation target. The animal has to acquire, look at the fixation target. This is the boundary that's where we track the eye position to make sure that the animal's maintaining its gaze. It maintains its gaze throughout the trial. And in one animal after a half a second, and in the second animal after 800 milliseconds, the central fixation target is replaced with one of 5 possible objects that are used randomly from a library of objects on a day-to-day basis. So the animal has a different set of stimuli on each recording session. That is presented for half a second. The animal has to keep its gaze within the boundary of the fixation window. So that's the sample period. And then that goes away. And then there is a variable delay where the fixation point comes back on. The animal has to maintain gaze, but also has to maintain a memory of the identity of the object that was presented. At the end of that variable delay, 2 objects are presented. One of them is the matching target, and one of them is a distractor. And in the second animal, they're presented at 8 possible locations on opposite sides of the visual field. And so a correct trial then would be simply all the animal would have to do is look at the correct object. And an incorrect trial would be if the animal looked at the non-matching object for the required amount of time. So that's the basic task. It's pretty simple. And we're really focused on looking at the patterns of the local patterns of activity in the local field potential. So here's a big part of the whole story. This is an example of one recording session. So what we've got here on the left is a line drawing taken from a photograph of the left hemisphere of the second monkey in our study. And each circle here is the location of the entry point of one of the electrodes in the array. And the first thing you notice is it's not the full array. So some of the circles are missing, and that's because the electrodes didn't work or they failed. So we had a subsample from the 256 channel array. And on any given day of a recording, which spanned 6 months in this animal, actually 9 months, excuse me, only a subset of the working electrodes were recording unit activity and field potential. So those are shown in black. And then the circles that are shown in blue are the ones that I've illustrated here, where you're looking at the raw broadband signal, the voltage signal coming from each electrode in, I forget how many of this is, something like 14, 14 different cortical areas. So what you see here is that we histologically, when the experiment was finished, histologically, we reconstructed all of the electrode tracks and identified the cortical area from the Markov map where every sample of data came from in our entire data set. So we were able to go back to each session and say, this signal came from area 2 of the somatosensory cortex. This signal here came from area 7A of the parietal cortex. And this signal came from area 946D, the dorsal portion of around the principal sulcus in the prefrontal cortex. So we had a very nice reconstruction, and that allowed us to look at the signal with respect to all of the different cortical areas that we made measurements from. And what you see here is the raw trace on a single trial. These are the vertical and horizontal eye positions. And at the end of the trial, the animal makes a short saccade to the correct target to get a juice reward. And the vertical lines here show the time course of the sample stimulus. And then the vertical line on this trial shows when the match stimulus appeared. And so you can see the animal maintains its visual gaze stably throughout the trial and makes a short saccade to the matching target. But what's really notable, and what is a large part of the story here, is that it's very easy to see by just visual inspection of this data that the temporal pattern and the frequency organization of the local field potential differs dramatically from one area of the cortex to the next. So in this case, in V1, there are large amplitude, slow frequency fluctuations. You move into extrastriate and parietal regions and you get a dominant, what you might call an alpha rhythm. Sure enough, in the somatomotor region, similar to what was originally reported by Jasper and Penfield, you have a clear beta rhythm in primary motor and primary somatosensory cortex. And then you get varying types of patterns of activity in the premotor and the prefrontal area. So for example, up here in area 946, the signal looks almost essentially flat, which has always surprised me. If you take and then what we did is, in order to analyze the data, we broke these signals up into different 400 millisecond windows. So we have a pre-sample period, we have a sample period here in S. D1 is the early delay period, D2 is the late delay period, and DM is what we call the match lock delay. So that allowed us to analyze the signals that occur just prior to the match, even though the duration of the trial was varying randomly across the trials. And then here in this plot is the power spectrum in linear coordinates. So basically amplitude as a function of frequency of the field potential signal in each of these 14 cortical areas. So these are the mean power spectra, and the different colors represent the different epochs of the task, right? So for example, black is the pre-sample period, green is the sample period, and so on. And so the first thing that you notice when you look at this is that the shape of the power spectrum and the location of the peaks in the spectrum vary from one area to the next, and they match nicely what you can see in the raw data. So for example, in V1, virtually all of the power is in the low frequencies and it falls off very sharply in the high frequencies. In area V6, you get a similar pattern, but there's a peak around 10 hertz. Area 2, there's a peak around 10 hertz. And these vary in amplitude. In the somatomotor regions, area F1 and area 3, you have this very strong beta component around 30 hertz, which you can see here in the raw data. This beta component extends up into the premotor areas and even into the prefrontal areas. It's not exactly shown in this plot, but I'll show you evidence for that. And then there are other things like shoulders here, and then small subsidiary peaks and alpha rhythm in premotor cortex and alpha rhythm in frontal eye fields. And so we decided that we were going to, in order to look at the spatial pattern of this more quantitatively, that we were going to extract some very simple parameters from these power spectra and plot them on flat maps of the cortex. And so this is a plot of a flat map of the cortex, a generic flat map illustrating the areas that we made recordings from. So this is the first monkey, this is the second monkey. In the first monkey, we recorded from a total of 39 areas, and some of the areas we made into small groups. We had 25 recording sessions, and in the second monkey, we recorded from a total of 58 areas, which some of them we combined, and we ended up with 33 groups of cortical areas. So for example, orbital prefrontal cortex, which is made up of multiple areas, if we had a small sample from each of those individual areas, we would group them together and call that orbital prefrontal cortex.
Charlie?
Yeah.
Can you go back one slide? On the panel C, you're showing the power spectrum for the different times of the task.
Right.
So was your expectation that those power spectrum that changed the most throughout the task will be more involved in the task, or what was the working hypothesis?
Yeah, it's so I'm going to end the presentation with a description of that. Hopefully I'll get to it.
Okay.
It's all rather complicated, but yeah, one would like to be able to focus and find out what's particularly relevant to visual memory. But interestingly, you can see right here at the very beginning of the presentation is that in area F1, there's a dramatic decrease in the beta rhythm. So it's strongly task dependent, but one would sort of argue that primary motor cortex is not likely to be this location in the brain where visual short-term memories are being represented. And yet shows a very strong task dependence. So
yeah,
very
that's
interesting. Yeah.
Okay, thanks.
Yeah, I'll try to get to that. It's a lot to present here. Okay, so the first thing we did was we wanted to find what are the appropriate frequency bands to look at. So we took every power spectrum from every recording in our entire sample, and we applied a simple peak detection algorithm to look for local peaks in the power spectrum. And then we accumulated all those peaks across all of the entire data set, 25 sessions in monkey 1. What was it? 62 sessions in monkey 2. And this is a histogram of the local peak frequencies in monkey 1 and the local peak frequencies in monkey 2, using the entire trial as the source data, not breaking it up into individual task epochs. And the very first thing that you notice is that these two monkeys have very different distributions of peak frequencies. So there's already a very interesting conclusion here, and that is that the frequency of narrowband oscillations in the cerebral cortex can vary significantly from one subject to the next. It's not always the same. And the bands, which are the minima between these distributions here, the bands that we chose, we had 4 bands in monkey 1 and 5 bands in monkey 2. The bands don't always fall into the canonical frequency bands that have been published for many years in the standard EEG literature, like theta, alpha, beta. So for example, this monkey has what we would call 2 beta components. The alpha component here is centered around 10 hertz, but in this monkey, the alpha component is centered around 14 hertz. And interestingly, this frequency here is not present in this animal. And we don't quite know why that is, but there's precedent for this result.
I mean, Charlie, I mean, this is very interesting because we have looked at many LFP from different animals, and it's true that even in rats you will find that they don't have the same features on different frequencies. So why an animal will have some frequency, or why one animal will have 14 hertz as the peak and the other will be 16 and the other, what doesn't mean those difference to you?
Well, certainly there's obvious reason to think that there's genetic differences. There's been some precedent for this in looking at the gamma band in identical twins and fraternal twins from the work of Pascal Fries. The animals have different genetic backgrounds and different environmental histories, and so they're going to develop differently. So it's a bit hand waving, right? But
but that doesn't mean that they confer them different capabilities of resolving cognitive question. I mean, it doesn't matter if it's 14 or 16.
Right.
You still do whatever you need to do, right?
Exactly.
So the actual frequency is not really relevant, or doesn't.
Yeah. I agree. And then if you look at the rough spatial distribution of these frequencies, the alpha activity looks to be widely distributed across the brain, but the different beta components are more frontally distributed. This band here, which would classically be called as part of the gamma band, we haven't demonstrated it, but we believe that this is a frequency harmonic of this strong beta signal in the second animal. Separate question to look at. So we derived some simple parameters from each power spectrum. We took what we call the spectral content, which is the ratio of the power in one band to the power across the entire spectrum. So it's the relative power within a band expressed as a percentage. We took the peak amplitude within each band. So for example, in the sample epoch in band 2, in this recording, the peak amplitude would be here. And then we used a separate measure of the time series that's referred to as sample entropy that we derived Stephen derived from the literature, which is the simplest way to describe it is the stochasticity of the signal, or the unpredictability of the signal, or the irregularity of the signal. And it yields a very nice metric. And then we can derive each of these parameters from the power spectrum or the time series for each of the different task epochs, and we can look at the task dependence. So the very first thing we did was we plotted the spectral content, the mean spectral content across all sessions in each of the areas that we made measurements from, where we had a sufficient sample. And you see a remarkable thing. So in the low frequencies, the highest amplitudes are in the occipital cortex and nearby areas. As you move higher in frequency, the distribution becomes more centrally located. And as you get into the beta band, you have a frontal distribution of the higher frequencies. And even though the frequencies differed somewhat from between the two animals, the same general pattern is occurring in the second animal, and even more clearly, right? So the low frequencies are distributed in the occipital region. So there's a gradient from occipital to prefrontal cortex. The alpha band is more central and perhaps somewhat frontally distributed. And then these beta bands are more frontally distributed. In this one unique band, the beta rhythm is highly localized to motor, premotor, and somatosensory regions, but it does extend a bit up into the prefrontal areas. And this has been known for a long time, but it hasn't been looked at such a global scale. So there's very interesting spatial gradients that the pattern of which depends on the frequency that you're looking at. So that's already a very interesting new finding as far as we're concerned, and it's a step above and beyond the old EEG methods. And then the sample entropy, amazingly enough, shows a similar type of gradient where what you would call the irregularity or unpredictability of the field potential signal is dominant up in the prefrontal cortex, and a very clear gradient up here in the second animal. So the signal seems to be quite irregular or stochastic or unpredictable in prefrontal regions compared to central, parietal, and occipital regions. So we can reach a couple conclusions from this. The frequency distribution of narrowband oscillations in the LFP can differ between subjects, and it doesn't always fall in classically defined canonical frequency bands. And the amplitude of the LFP exhibits distinct spatial gradients that depend upon the frequency of the fluctuations. And the entropy of the signal, or the irregularity of the cortical LFP, exhibits a strong occipitofrontal gradient that peaks in the prefrontal cortex. And we can discuss why that might be. So then we want to know, are there any underlying anatomical correlates for these gradients other than the basic cytoarchitecture? And this is a bit of a side step, but I'll try to make it quick. There's a body of work that's done by this person named Guy Elliston that I recently discovered from reading some other papers, and then went back and looked carefully at it. And it's a beautiful body of neuroanatomy. And essentially what they did was in a large number of studies spanning about 15 years, they would take pieces of cortex out of different parts of the macaque monkey brain, and they would inject the cells with lucifer yellow and then stain the cells, and then do a classic analysis of the dendritic field size of the neurons. And they had large numbers of neurons that they did this in many, many different cortical areas of the brain. And it's published in a whole series of studies. But this particular one shows that the dendritic field size shows a nice gradient going from V1 to V2 to V4 to MT to TE, and then into the temporal lobe. So the cell size, the dendritic field size dramatically increases as you move up the visual hierarchy. And that's shown in this plot here, which is the basal dendritic field area of, in this case, layer 3 pyramidal neurons. And then they went above and beyond this, and they counted the number of dendritic spines, which directly reflect the number of excitatory synapses onto the dendrites. And they found that the density of synaptic spines increases along with the size of the dendritic field. So there's a very big difference in the total spine count on cortical neurons as you move up the cortical hierarchy. And they essentially counted the basal dendritic spines in layer 3 pyramidal neurons in visual areas. They did it in prefrontal cortical areas, and they did it in somatomotor areas here. And one of the nice quotes from this beautiful review article in 2007 is that there's a consistent and dramatic increase in the number of spines in the dendritic trees of pyramidal cells as a function of the absolute distance from the occipital pole. There's a striking difference here. And then in this study by Chaudhuri out of Xiaojing Wang's lab back in 2015, they took the hierarchical measurement that Markov had developed, Markov and Henry Kennedy, based on the ratio of superficial and infragranular cells of origin. And they compared that to the spine count of the cells in each of the cortical areas, and they found this remarkable correlation. I mean, just striking correlation between position in the hierarchy and the number of spines on the dendrites of the pyramidal cells. So, of course, having measured these gradients, we wanted to see if something similar was going on in our data, and we found some evidence for it. If we looked at the spectral content over a set of 14 or 18 areas in the two animals, we looked at the spine count taken from these publications and the spectral content in the low frequencies, we found a negative correlation. So the higher the spine count, the lower the amplitude of the low frequency components. And I was particularly interested in this one because of the sample entropy showed a very nice gradient going from occipital to prefrontal areas. And as the spine count increases, moving anterior in the brain, there's a correlation with the gradient with the sample entropy. So this indicates there's a very nice, an additional anatomical correlation with the dendritic field size and the density and count of excitatory synapses on the basal dendrites of pyramidal neurons. So then we thought, okay, we have these very interesting gradients. Clearly, there are differences in the spectral properties and temporal properties of the field potential in cortical areas. There's an anatomical correlation. We wanted to know if we could decode cortical areas on the basis of these spectral features. And then we wanted to know how the spectral features vary with the task. The spectral properties of the LFP vary with the task. So two big questions, and I'll try to get through this. So we applied a simple support vector machine analysis to decode cortical areas. We used the spectral content, the peak amplitude, and the sample entropy in each of the 5 task epochs: pre-sample, sample, early delay, late delay, match lock delay. And we only used data from areas in which we had a minimum of 20 recordings. And when we had more than 20 recordings, we would subsample the data. So we always had the same sample size that went into each analysis. And so this reduced the total number of areas in monkey 1 to 11 cortical groups, 11 areas or groups in 4 different frequency bands, which gave us 9 variables to look at. And then in monkey 2, we had a larger data set. So we had 27 cortical areas in 5 frequency bands, which gave us 11 variables for each epoch. We used a k-fold cross-validation scheme where we would train the data on 80% of the trials and then apply the analysis to the remaining 20%. And we would repeat that analysis 1,000 times with a random sample of 20 recordings on each iteration. So we would get a distribution of measurements that we could then look at get a stable estimate there. And we would run the same calculation, but randomize the assignment of cortical areas. So we had a measure of a surrogate, a random surrogate. And so the chance performance on this analysis for the first animal with 11 areas was 9%. So we would expect that we would guess correctly randomly. If the data were random, we would guess correctly 9% of the time where the signal was coming from in the cortex. And in the second monkey with 27 areas, the random guess rate would be around 4%. So very low if the data were random. So this shows the confusion matrices, the mean of the confusion matrices across all of the analysis for 11 areas in monkey 1. And so the diagonal of this represents the number of correct choices that we made, right? So we randomly pick a field potential recording from one cortical area, say 7B, and roughly 60-70% of the time we identified that area correctly as coming from area 7B. So all of the data points off of the diagonal represent errors or incorrect assignments. So we refer to that as validation accuracy. But the real data are really down here, which is the plot of the diagonal values in the medians here in red. And then these are the distributions of the results of the 1,000 iterations. And then the red line here is the 99% confidence interval derived from the surrogate distributions. And what you can see is that every cortical area was well above chance. Some were a little bit closer to the 99th percentile, but in general, we were very good, well above chance here, and clearly above the confidence interval of being able to decode the cortical areas. So this clearly indicates that the power spectrum in the different bands, or the amplitude in the different bands, and the structure of the time series derived from the sample entropy gives you enough information just with 3 simple parameters looking at different epochs of the task to reliably decode the cortical area with pretty high confidence. And then a short aside, we applied the same analysis to the other epochs of the task. And so these plots here show the diagonal values here in each row, color-coded to the validation accuracy here with respect to the different epochs of the task. And I'll just tell you very briefly, this is kind of a side point, but every single area had a different validation accuracy as a function of the epochs of the task. So there was clearly task dependence in the decoding, but this is not really the appropriate analysis to look at that type of question. So the punchline here is really that you can decode cortical areas. Their spectra are truly different across the cortex. So then regarding the task dependence, which gets to your question, Pedro, the decoding analysis really doesn't give you a useful way to analyze that. So we went back to a much simpler method, and that was simply to take the power spectrum on each epoch of the task, on each trial, and to calculate the mean value in the power spectrum, or the mean amplitude within each band, right? And so we would compare the sample epoch. We would compare each of the epochs of the task, the sample, the early delay, the late delay, and the match lock delay. We would statistically compare that to the amplitude in the pre-sample epoch, and then look to see if there was a statistically significant difference. And so in this example recording from area 8B in band 2, which is the alpha band, we had 60 recordings. In this case, the second animal. And out of that, each of these colored circles here is one recording that was statistically significantly different from the pre-sample period. And the black dot indicates the mean of the distribution of the significant recordings, and the horizontal line indicates the incidence of significant differences. And so one thing you see in this example is that during the sample period, all of the significant cases are increases in amplitude compared to the pre-sample. The incidence of that occurrence is about 25%, about 40-45%. and the amplitude is about 25% larger. When you look at the early delay and the late delay, you see a mixture of increases and decreases in amplitude. And the magnitude of that change on average and the incidence also varies as you move through the task. And then in area F2, which is a premotor area, every single recording, almost every single recording, nearly 100% of the recordings, showed a statistically significant suppression, which increased in amplitude throughout the task. So we did a simple way to try to remap this for all the data. We color-coded the amplitude of the change. This is the black dot. And we color-coded the incidence of the change, in other words, how often it occurred, by just plotting these graphs here. So you can see, based on these color codes here, that this was the mean value of the increase in amplitude with each epoch, and this is the mean value of the decrease in amplitude with each epoch. And so you can see there's a mixture of increases and decreases, and then this is the incidence, or how often those changes occur. And you can see that here in F2, there was an increase in the magnitude of suppression, and nearly every recording, almost every recording, showed a statistically significant suppression. Okay, so that's quite a handful. And then this next one, which is the final result I'll show you, is basically looking at these plots here across all cortical areas in both animals. So it's quite a handful here, but this is the basic spatial pattern of task-dependent changes in the LFP spectral power as a function of the epoch compared to the pre-sample period. These data show the amplitude changes for significant changes. And here's the scale for that. And these data here show the incidence of occurrence of statistically significant increases and decreases. So it's pretty difficult to summarize all this, but a couple points are worth noting. The very first one, and I think the most notable result here, is that every cortical area has at least one band or one epoch, most of the time multiple bands and multiple epochs, where there is a statistically significant change in the LFP power compared to the pre-sample period. And so the really simple conclusion from this is that every cortical area that we measured in both animals showed some task dependence in this very simple task. And that I think is probably the most significant result here from this certain early analysis. And that is that every cortical area is involved in some way. It's showing some type of response to this very simple overtrained task of simply holding, perceiving, and then holding in short-term memory a simple visual object that's been overtrained, highly overtrained. So task-dependent changes are widely distributed across the brain. Which ones are critically involved in the performance of the task yet remains to be seen, but you can see some interesting things here in the incidence. So in prefrontal cortex, there's an increase in the incidence of increases in power, right? When you look at the higher frequency bands, there's a profound suppression of the beta component here. And there's some variations between animals, and probably the clearest result is in the second animal. But and then there's some cases where you have what look like very tantalizing effects in some extrastriate areas, in prefrontal areas that might be particularly relevant for representing the stimulus. But the bottom line is that these changes are all essentially all over the brain, widely distributed. So we can reach a few conclusions here. Cortical areas can be reliably decoded from a small number of simple spectral and temporal features in the LFP. During a simple visual mnemonic task, such as holding one object in memory for a couple of seconds or even 1 second, all of the cortical areas that we made measurements in exhibited some change in the spectral power. that varied in the magnitude and the sign and the frequency and the incidence of occurrence. So there's widely distributed changes. And you can sort of reach a few simple conclusions. The spectral organization of the LFP is globally structured. So there are these gradients, but within each area, there's a high degree of local heterogeneity. So there are variations in the frequency and the amplitude of the signals, and their task dependencies are also heterogeneous. So it's complex, to make a long story short. And I think we can, with this finding, especially in combination with the results of Elston, the synaptic analysis, I think we can pretty confidently state that Mountcastle was wrong. The different areas of cortex do exhibit qualitatively distinct modes of intrinsic operation. So the basic dynamics of each cortical area are different. So this would imply that models of cortex that assume that the module, the cortical module or the column of cortex is the same everywhere in the brain, I think are essentially qualitatively incorrect. That's one conclusion, I think, a global conclusion we can reach from this. So I'll end it there, and thanks a lot. Really appreciate it. Happy to take questions.
Thank you very much, Charlie.
Yeah, I have a few questions, but I would like to, there's applause all over the place.
Thank you. Muchas gracias.
You cannot hear them, of course, but before I do have some questions, I will just have let somebody else's ask questions, or I'm not see hands raised yet, but I will break the ice, maybe. And so this some things I'm not quite sure I understand, because I had the impression that maybe a particular task like this one, the oscillatory activity will, rise simultaneously in many places because of, functional communication. Mm-hmm. But it seems that you're showing by demonstrated that you can decode things, that this may not be that often, because if you have the same large oscillations everywhere, you will not be able to decode it. So it seems that if it's tumor homogeneity, you cannot discriminate.
Right.
So how come, what happened then with the idea that when you're doing a task, you just, every cortex involved gets the same oscillatory activity? And so there seems to be something that's not,
The simple answer to that is we can reject that. We can reject that idea. That's not what's happening. Right.
Wow,
So
that's a big change. Yeah. I need to change what I'm teaching.
There's no globally structured rhythm that is some type of information carrier, for example. I think we can reject that idea. And it will depend on some future analyses of the coherence, for example. But I think we can state that different areas are doing different things during the task. There's no one particular frequency band that's necessarily doing more than another, and some frequency bands are strongly suppressed. We don't know what the meaning is of some of these results. All we can do is describe them, but I think we can sort of dispel the idea that there is a global, information carrier that's common among the regions that are intimately involved in the task. If there were any one particular rhythm that I might choose to look at, I would probably focus on the alpha rhythm, because it does seem to be widely distributed. And there's some very interesting preliminary findings we're getting from the coherence analysis, but I won't go into that.
Okay, thanks, Pablo, and then Jimena. Your microphone is muted.
Thanks, Charlie, for the great talk. I would like to ask you if it's possible to relate the electrical activity or spectral activity that you show in different areas with some particular feature of the working memory or different contents that
Yeah, so a lot of that analysis we have not done because we kind of had our hands full with all of these other variables. But now is the time to go look more carefully. So, for example, the task dependence that I showed you, that's averaged across all the stimuli, right? And on each session, there are 5 stimuli. So there's good reason to think that the task dependence will differ from one stimulus to the next. And that's another question that we need to look at. But it gets complicated, right? Because you have all these areas, many different recordings in each area, and then you add 5 different stimuli, and you have, the choice of the animal, correct and incorrect. So it's a complicated analysis, but we're going to look at that. And unfortunately, I don't have any real strong conclusions to tell you about that. We're basically at the stage of saying, well, we see task dependence essentially everywhere. We don't know which of these task dependencies are critical for the task. And it's something that we still have yet to look at. We did publish a paper from recordings in parietal and prefrontal cortex back in 2012, where we looked at the stimulus dependence of the coherence. And we did find that there were selective patterns of coherence between prefrontal and parietal areas that depended upon the stimulus that was being held in memory. So we have good reason to believe that there are distinct spatiotemporal, circuits that represent the different stimuli, and that they differ quantitatively from one another. But we haven't gone down that path yet. We did look at it with respect to unit activity, and we found a very interesting set of results in the unit activity, which we published in a 2018 paper. So I could refer you to that result there. And yeah, there's a lot of task dependence in the unit activity, and the stimulus specificity seems to be more restricted to association areas of cortex, such as parietal and prefrontal. Even though all these other areas show some task involvement.
Okay, great. You still have your hand raised, Pablo, but it seems that you... Okay, Jimena?
Oh, okay, thank you. Thank you very much for the seminar.
Sure.
It was great. I'm not really a cognitive neuroscientist, so of course my focus was in this incredible difference between the size of the cells in different cortical areas, and also the amount of synapses. Well, the amount of excitatory synapses.
Yeah.
So you also showed the correlation between the activity of these areas with the number of synapses. Do you think that just that, I mean, just the number of synapses is the difference between the function or the LFP gradient,
Yeah.
I mean, there are also many other differences, but do you think that that feature is really the difference between the
Well, of course, different cells have different intrinsic membrane currents, right? So there could be, there's evidence for gradients in the different types of inhibition across the cortex. There's evidence for different intrinsic membrane currents in different cell types across the cortex. And I would refer you to a paper from Xiaojing Wang, a review paper. I want to say it was Nature Reviews Neuroscience, but it's from 2020, where he looks at a lot of these different anatomical and physiological features. And that is where I actually got the idea to do this analysis, because I find it absolutely amazing that the number of synapses on a V1 neuron it's 10 times greater in the prefrontal cortex, a factor of 10. and the dendritic field size.
Amazing.
And so the way that the neurons in prefrontal cortex are integrating information, they're also getting input from a much larger number of cortical areas. So they need to somehow have mechanisms to select which of the synaptic inputs that they're getting are the ones that are going to be processed. And so this produces a great degree of heterogeneity in the synaptic input onto these neurons. And I think it's directly responsible for the greater degree of sample entropy in the LFP. In other words, the LFP is desynchronized because the pattern of synaptic input coming into those areas is highly heterogeneous. And it's being selected and switched dynamically in these neurons. And I think it's a key feature. And I'll show you one really tantalizing result here. If you let the animal fall asleep, the characteristic onset of sleep is what's called a sleep spindle, right? And so this is the alert state here, right? And just as the animal starts to fall asleep and close its eyes, you get this very strong
synchronization.
And so we know that the neurons up in prefrontal cortex can become strongly synchronized. But in the normal vigilant state, awake state, they're highly desynchronized. And that's probably what's giving rise to this increase in sample entropy and the lower amplitude, right? But clearly the gradients that I was describing are going to be strongly state dependent. So the gradients that I showed you are very, under a very set of very special circumstances. And so here in this state, all of the synaptic input coming into those prefrontal neurons is highly synchronized, and they're engaged in this profound rhythm that happens during sleep. And so, I think the synaptic anatomy is absolutely fascinating.
Thank you.
Thank you, Jimena. Vicente has a question, maybe the last one, given the time.
Yes, thank you very much. Thank you, Charles, for the amazing presentation and also amazing data set. It has to be noted that the data set is very, very nice.
Thank you.
I just wanted to maybe move a bit the literature towards more macro scale neuroimaging, particularly in fMRI. Most of the studies studying gradients in the brain. I'm particularly talking here about the work from Dan Margulis, has shown that there are functional gradients that are in resting state, but they're also reconfiguring when you're in a task state. And also a very interesting discovery a while ago was that these functional gradients are supported also by the structural connectome of the brain. Do you think that the structural connectome as measured by, for example, tractography or something like this could be a source of variance of this type of spectral heterogeneity we are seeing in LFP?
Oh, absolutely. Absolutely. So if you look at this early result that I showed, again, this is in the monkey, but we have no reason to believe that something similar is happening in the human. And in fact, the work so this would indicate that regions in prefrontal cortex are getting input from many more cortical areas than early sensory areas, right? And there's a direct correlation between this and the number of synapses on the dendritic fields of the pyramidal neurons, right? And Elston, actually, if you look at that work, I would really recommend looking at it, because I was unaware of this beautiful neuroanatomy until basically a year ago. And then it was a revelation to me to see this. And because I had always believed in sort of the Mountcastle model, right, that a cortical column is a cortical column no matter where you look at it. And I was just kind of a blind neurophysiologist, And then looking at this beautiful neuroanatomy, I realized, well, that's absolutely not the case. And Elston makes this case beautifully. And then with the connectome, however it may be derived, whether it's through through imaging or track tracing, the more specific results would be from the track tracing. Strongly supports these types of gradients, these anatomical gradients, and
So
mapping
the interesting thing now, like the probably the next challenge would be what neural process is making the gradient change over the variability already explained by the connectome, right? So you have the connectome that is giving a certain gradient, but also the functional activity over that connectome is changing depending on if it is resting state or task state, so on. So the question will be what neural process is changing over the already explained gradient by the connectome. So
absolutely. And this pattern that I showed here, just in the raw data here, this is one snapshot, right, of what the data looks like in one monkey. And if the task changes, from a visual memory task to sequence memory or some type of selective attention task, any one of a number of things, you could expect unique changes in this activity, depending upon task and the level of arousal and vigilance and so forth. So this is an initial snapshot of what's going on. One big mystery is why is the temporal structure so different in different areas? And I think the synaptic connectivity explains part of it, but the intrinsic physiological properties of the cells and the circuits in each of these areas is going to be different as well. That can explain part of that. So, the old Mountcastle view here is looking a little bit weaker than it used to.
Could it be that the cortical columns are just increasing in size as the dendritic trees?
Well, the cortical columns are defined by this phenomena called a mini column, right? Which probably has a developmental, origin. And I can't speak very, very intelligently about that, but there's something dramatically different about basic cellular anatomy here, from one area to the next, right? So not only does the dendritic field size increase dramatically, right? Like fourfold, but even the density of the spines of the auditory synaptic input is increasing. So when you take the combination of the increased dendritic field size and the increased synaptic density, the count of the number of synapses is a factor of 10.
Wow.
Factor of 10. And one of the studies by Elston, I think it's 2011, they compared it to different species of animals. And I think this basic gradient, I think there was a claim in that study that this gradient was not present in rodents, but it was strongly present in humans, and the largest number of synapses that occur in the cerebral cortex were in the prefrontal cortical areas of humans. So
there's a very new paper, 1920, by Mark Harnett, that they show actually the biophysical properties of human cells are very different from any other animal.
Wow.
But also, I was trying to point out that also, columns are defined typically by the receptive field properties, which may work very well for early sensory cortices, but no way from other cortices like prefrontal cortex. So there may be something fundamentally different in there.
I think that the notion that neurons in the same as you and I worked on, neurons have the same receptive field properties is really something happening in early sensory areas, because even I've talked to many people that study motor cortex, that even in motor cortex, there's even more heterogeneity among adjacent neurons in motor cortex. And then in prefrontal cortex, I would make the prediction here that adjacent cells are doing probably quite different things in
Right.
association areas.
I was completely blown away by your figure where you showed that F1 has the largest change in LFP spectrum. And that seems to tell us that motor cortex is not only motor cortex, but it seems to be heavily involved in many processes that are not look like motor
Exactly.
processes. And maybe because we evolved as animals that move around, motor cortex always was part of every activity, even cognitive activity. So
yeah,
very cool, very cool.
Yeah, I agree.
I wonder whether, let me check whether we're almost done, so we have no more time. So Alberto, maybe you have one step. Okay, so I would like to end this thanking Charlie, because I didn't say that at the beginning, but he was my postdoc advisor, and I couldn't have made a better choice to go and learn with him. So I think a lot of what I've done through the years is led with him. So thank you, Charlie.
Oh yeah,
and thank you for the talk.
Yeah, thank you very much.
Okay, so thank you everybody for coming, and have a good summer. And I think Pablo is not another seminar, I guess, before next year, right? Okay, so thank you everybody, and see you next year.
Okay.
Okay,
Bye-bye.
thank you. Bye.